We need to test soil around peonies specifically to determine the nutrient content within the soil that the plants could potentially be using later on during their growing season. Soil testing alone will not provide the total answer for plant growth. You need to correlate soil testing along with plant tissue testing to see how much the plant is actually taking up during the growing season. Certain factors related to the growing conditions and nutrient uptake that the grower has no control over would be the weather, would be temperature, would be rainfall, soil moisture, things of that sort. Each of those are going to be an important aspect on how nutrients are converted from inorganic sources into mineral sources that can then be taken up by the plant and used later. Other methods other than nutrient analysis should include something along the lines of available pH. Nutrient uptake is going to be impacted greatly by the acidity or the alkalinity of your soil. The organic matter content is either going to tell you how much is going to potentially become available or how much is actually going to become unavailable and tied up by the microbes as they break down that organic matter content. So all of those things are important to have within your analysis. The nutrient analysis done within plant tissue shows the level of the nutrients within the plant at the point in time that it was sampled. Those nutrients are important then to reach whatever physiologic growth stage that you're looking for. And in peonies, obviously the end physiologic growth stage is that nice big huge flower. So you're going to want to take the sample prior to it actually flowering to see whether or not you have enough nutrients in the plant to reach that flowering stage. Plant tissue samples do vary by point where they're collected in the plant. For example, if you have a growing plant, most of the nutrients will be in the growing tips, in the top stems, in the outside leaves, that sort of thing. As the plant reaches much more closer to maturity, then the nutrients are going to start to be translocated to the reproductive parts. So they're going to go to the flowers, they're going to go to the roots, which become next year's plants, and you're going to wind up losing a certain amount of nutrients from the stems and the leaves. Research protocols for existing crops, both from an agronomic standpoint and from a horticultural standpoint, are long known. They are related to specific plant parts at specific growth stages. They relate to the production of either biomass or flowers or seeds and the maintaining a maximum quantity and quality of these products. You can find all sorts of graphs and charts that have high and low ranges for nutrients within the soil and in the plant part that can then give the grower a better idea or a better understanding of how well nutrient management is working for him and his crop. For peonies, that doesn't exist, especially in Alaska, and so that's what we're trying to accomplish with this study. Tissue samples will allow us to get a better insight in how the soil nutrients have been taken up by that particular plant. Obviously, soil samples alone aren't going to work. You're going to have enough plant tissue samples that go along with that, collected at the exact same time, to see whether or not that plant actually has the sufficient amount of nutrients it needs to reach the flowering or the reproductive stage, whatever you're looking for in this particular case. The research goal for this entire study is to establish that range that was talked about a little bit before. We need to find out what the good and the bad levels are within the soil, what good and bad levels are within the plant tissue, so that the grower can then have his plants and soil samples analyzed and know where he falls within that range. That's not going to provide you with an immediate management technique for this year, but it will provide information for next year's management.
In order to establish an acceptable range of plant nutrients, we need to collect tissue samples from plants that look healthy with large green leaves, good growth, and collect leaves from plants that don't look as good with pale yellow leaves, poor growth, to compare against. Both sets of leaves should be collected from plants that are as close to the same growth stage as possible. In this case, peonies should be at a bud maturity index of 2.5, where a single petal separates at the top, exposing the flower, and the bud itself is still hard. The plant part that needs to be collected will be the first set of leaves, plus the petiole directly beneath the bud. You can remove the leaves from the plant with a sharp knife or a pair of scissors. Do not take more than one set of leaves from any plant, so you don't stress subsequent plant growth any more than you already have. In order to have enough dried plant tissue for analysis back in the lab, you'll need to collect at least 4 samples. Each set of leaves should be collected from 4 individual plants. Clean any obvious signs of dirt and insects from the leaves, and make sure they are as dry as possible by blotting them with a clean paper towel. They can be put in the same paper bag as a composite sample to send off for analysis. When collecting leaves for plant tissue analysis, it's important to avoid as much bias as you can. Try to avoid high traffic areas, edges or ends of beds or rows, fence lines, and definitely any plants that have been recently treated with a foliar fertilizer or pesticide. Sampling representative plants from the middle of a row or a bed is best. When sampling poor growth plants, try to avoid obvious diseased plants if possible. Collected leaves are put in the same paper bag. Look for the number grade printed on the bottom of the bag. Paper bags that are labeled number 8 through number 14 work best for sample collection. You should have 2 paper bags, one with good growth plants and one for the poor growth plants. Don't ever put plant tissue in a sealed plastic bag, as biological processes will continue, leading to the beginning of decomposition. This will change the nutrient status of the tissue dramatically. Paper bags will allow for some moisture loss, slowing down but not stopping the decomposition process. Even so, the plant tissue samples need to be dried as soon as possible. So mail them off for continued drying and preparation after collection. The bags need to be labeled with the following information: your name, your farm, your address, the date of sampling, the location of sampling, that's the bed, row, field, plant number, or anything that will identify where each sample came from. The plant variety or cultivar name, if sampling more than one variety, each will have separate bags, and good or poor plant. In order to correlate the soil samples with the plant tissue samples, we need to establish an acceptable range of soil nutrients by collecting soil samples from plants that look healthy with large dark green leaves, good growth, and collecting samples from plants that don't look as good with pale yellow leaves or poor growth to compare against. You'll only need a 1 inch by 1 inch by 6 inch deep sample. If you have access to a soil sampling probe that has the depth previously marked, then this is a quick and easy process. If you have a trowel, then it gets a little more complicated as you need to dig out a hole first and then collect a sample. It's best if you have a trowel that has a depth measure on the blade. Sweep away any recently deposited plant and organic matter from the surface of the soil. That's the easy part. The difficult part is collecting a sample as close to the base of the peony plant as you can without disturbing the major roots. Most of the original fertilizer additions were added to the same hole as the root at planting, and any subsequent fertilizer additions were most likely added to the soil surface near the base of the same plant. This soil nutrient reserve is why we need to get soil samples from as close to the plant as possible. Established peonies can have root systems that extend as much as 2 feet in any direction, which is why care must be taken when sampling. Starting a few inches away from the base of the plant, on any side, slowly insert the probe or trowel with just enough pressure to penetrate the soil surface. If you feel resistance, stop, as you may have encountered a major root. Move over a few inches and try again. Once you've reached a 6-inch depth with the probe, remove it from the soil and slide the soil sample out and into a 1-quart size zipper-type plastic bag. If you are using a trowel, dig a hole large enough for you to see the back side. Then slice a 1-inch soil sample from the back of the hole. Remove it and clean off both sides until there is just a 1-inch by 1 inch by 6 inch deep sample left on the blade. Slide that into the plastic bag. Repeat this process on the other side of the plant and again for the remaining plants. Combine all the soil samples in the same zipper-type plastic bag. You should have 2 plastic bags, one with the good growth soil and one for the poor growth soil. Here again, moist samples in a sealed plastic bag will continue the biological processes. This will change the nutrient status of the soil dramatically. So, the soil samples need to be dried as soon as possible. Leave the bags open to aerate until you're ready to send them to the lab. Mail them off for continued drying and preparation as soon as possible after the collection. The bags need to be labeled with the following information: your name, your farm, your address, the date of sampling, the location of sampling, which includes the bed, row, field, plant number, or anything else that will identify where the sample came from, the plant variety or cultivar name. If sampling more than one variety, each will have a separate bag, and good or poor plant. For plant tissue analysis, you'll want a lab that will do the following standard plant tissue analysis: nitrogen, phosphorus, potassium, calcium, magnesium, plus the trace elements boron, iron, manganese, copper, zinc, and aluminum, as well as determine the elemental ratios of the major nutrients. The lab will need to know the plant type, what part of the plant was sampled, and where on the plant the samples originated from. In this case, the information would be the uppermost, full set of leaves from the peonies. They will also want to know if the sample came from a normal or good growth plant or an abnormal or poor growth plant. For soil analysis, the lab should be able to do the following soil sample analysis: active pH, SMP Socorro buffer pH for lime additions, percent organic matter, estimated nitrogen release, Bray 2 test for phosphorus on acidic soils, available NH4 ammonium and NO3 nitrate, total cation exchange capacity, percent base cation saturation, KCl extractable aluminum, and the Melic 3 extractable phosphorus, manganese, zinc, copper, iron, aluminum, sulfur, calcium, magnesium, potassium, and sodium. Be sure to inform the lab that all the soil samples came from a 0 to 6 inch depth. There are a number of ways the data can be presented to you after analysis. You'll want to be sure to get it in a couple of ways: in parts per million and in pounds per 1,000 square feet. Samples should be sent to a laboratory that does both plant tissue and soil testing analysis. UAF does not do the analysis, but contact a UAF professor of agronomy and soil sciences or the Agriculture and Forestry Experiment Station research technician for technical advice and contact information and mailing addresses for laboratories that do standardized testing.